Today I am going to discuss about one of the most important chapter, one topic, a neat topic, Plasmodium vivox life cycle. Here the classification, it belongs to phylum Protozoa, subphylum Epicomplexa, class Telosporia, and genus Plasmodium, and the species vivox. Yeah, commonly we know that Plasmodium which causes malaria. Here I am going to explain about the history of malaria. Malaria, it's an Italian word which has name as meaning is mal means bad, area which means surrounding. Here in early years they think that the malaria may be caused by the intaking of bad air. Later on, the first time Sir Patrick Monsoon he suggested that the malaria may be caused by the mosquitoes. Then later on, Charles Laveran he identified the Plasmodium vivox, Plasmodium in the malaria patient blood. Whereas Sir Ronald Ross he is a British scientist, he observed the oocyst of Plasmodium vivox in the female Anopheles mosquito. Here hence he is awarded by the Nobel Prize. Here there are the four major species which may causes the malaria. The first thing is Plasmodium vivox and Plasmodium falciparum and Plasmodium ovale, Plasmodium malariae. Here these are the four major species which causes the malaria to human beings. Here actually the malarian parasite, it's a digenetic parasite, which mean di means two, which mean the completion of their asexual and sexual maturation in a two host is also known as digenetic parasite. What is mean digenetic parasite? Which mean here which the parasite complete their asexual and sexual maturation in the two host is also known as digenetic parasite. Here whereas in Plasmodium vivox this asexual reproduction takes place in the asexual reproduction which takes place in the man, and sexual reproduction takes place in the female Anopheles mosquito. It's a digenetic and intracellular parasite in man, whereas in mosquito it's an extracellular parasite. Here intracellular parasite, which mean here the parasite which lives inside the host cell is also known as intracellular. Extracellular mean the parasite which lives at the outside of the host cell is also known as extracellular. Intracellular in man and extracellular in the female Anopheles mosquito. Here now I am going to explain the asexual reproduction of Plasmodium vivax in man. Here the sporozoids are the infective stages of Plasmodium vivax. Here the sporozoids are sickle shaped structure. At the anterior end which consists of a small depression which also known as apical cup, and a pair of secretory organelles which has opens in it. Here these secretions are helpful for the penetration of liver cells during hepatic schizogamy, and in the cytoplasm which consists of small filamentous tubules are called as microtubules, and the cytoplasm containing one nucleus and the other convoluted tubes also present in the cytoplasm. These convoluted tubes function till is unknown, but whereas the microtubules are helpful for the give the wriggling movement of the sporozoid. Here according to Bano, when a infectious mosquito bites a person, nearly 2000 sporozoids are enters into human being body. Within half an hour they reach to the liver cells. Here the asexual reproduction of Plasmodium which complete in two cells. The first one is hepatic cells and the second one is erythrocytes, or it is also called as RBC. Here generally the asexual reproduction of Plasmodium is also known as schizogamy. Here whether the asexual reproduction which has takes place in the hepatic cells then named as hepatic schizogamy, whereas the asexual reproduction in the RBC cell they are known as erythrocytic schizogamy. Again the hepatic schizogamy which consists of two cycles. The first one is pre-erythrocytic cycle and the second one is exoerythrocytic cycle. Let us, I am going to draw one diagram to explain the pre-erythrocytic cycle and exoerythrocytic cycle. The first, when an infectious mosquito bites a person, within half an hour the sickle shaped sporozoids are enters into the human being body. Through the blood it reaches to the liver cells. Then they are modified into a feeding stage known as tropozoid. It attains, it feeds the food materials which are present in the liver cells, and it increases their optimum size. Then later their nucleus which has divided into many successive mitotic divisions. Then after the karyokinesis, the cytoplasm also which has divides into each bit of nucleus. Then later they are modified as Scheidt stage. When the rupture of liver cells which produce cryptozoids, the formation of cryptozoids, up to formation of cryptozoid, they are known as pre-erythrocytic cycle. When the sporozoids are enters into liver cells, they are modified as tropozoid. During the tropozoid stage, they feeds the nutrients which are present in the liver cells, and it increases their optimum size. Then later the nucleus of tropozoid which has undergoes many successive mitotic divisions. Then later when they are modified into Scheidt, when the rupture of liver cells which produce cryptozoids. Here the cryptozoids which have two options, two pathways. To increase their number, again they attacks on fresh liver cells. Then what are the steps are observed in the pre-erythrocytic cycle? The same in the exoerythrocytic cycle also observed. First, they are modified, the cryptozoids are modified into tropozoid stage. Then later these tropozoid stage are modified as Scheidt. When the rupture of liver cells which produce two kinds of second generation of cryptozoids, or it is also known as metacryptozoids. Here the metacryptozoids are two types. The smaller, they are known as micro metacryptozoids, and the larger, they are known as macro metacryptozoids. Here the micro metacryptozoids always attacks the fresh RBC cell. Macro metacryptozoids always attacks on the fresh liver cells. Another way of the cryptozoids is they attacks on the RBC to start the erythrocytic zygogamy. Now I am going to explain the erythrocytic cycle. Whereas the erythrocytic cycle which initiate with the help of micro metacryptozoids or with the help of cryptozoids. Here these are enters into the fresh RBC cells. Then they are modified as tropozoid stage, increases their size, and it feeds whatever the food materials are present inside the RBC cell. Then later they develop pseudopodias around their body. At this stage is also known as amoeboid stage. Then later the pseudopodias are disappeared and they develops a vacuole, pushing of vacuole into peripherally which form a ring shaped structure, hence they are named as signet ring stage. Then later the vacuole will be disappear and the nucleus of the parasite which has undergoes many successive mitotic division which form schizont. After the nuclearic division, the cytoplasm also divides into each bit of nucleus. Then later nearly 12 to 16 merozoids are arranged in the petals of rose like this. At this stage is also known as rosette stage. Rosette stage. Then later the volume of RBC cell which get increase. These parasite they converts the hemoglobin into heme and as well as the globulin. The heme part which has converted into hemozoin, unsoluble crystalline hemozoin, and the globulin part which has ingested by the parasite. When the when the volume of the RBC which has increases optimally, the RBC cell which get rupture. When the rupture of RBC cell which produce heme and as well as the merozoids, heme and as well as merozoids. When the hemozoin which has releases into the human being bloodstream, then an organism which has get a fever. Here prophylaxis methods, how we can control the malaria. First of all, using mosquito nets and mosquito coils, and also culturing of gambusia fishes in the stagnant water. Here the gambusia fishes is a larvivorous fishes, they feeds the larval forms of the mosquito. And whereas the spraying of petroleum substances in the stagnant water and the rearing of insectivorous plants around the stagnant water. With the help of this prophylaxis method, we can control the malaria. Here the repeated division of the merozoids, some of the merozoids are reached into the spleen and as well as bone marrow, and they are developed into male gametocyte and female gametocytes. The male and female gametocytes are circulated in the malaria patient blood. When a female Anopheles mosquito sucks the blood from the malaria patient, the male and female gametocytes and other stages of Plasmodium which has reached into the mosquito's crop wall, the other stages of Plasmodium which get digested in the crop, and whereas the male gametocyte and the female gametocytes go for the sexual reproduction. The sexual reproduction takes place in the female mosquito. In the next video, I will tell you about the sexual reproduction of Plasmodium vivax. Thank you.